All right. Thank you all for joining today. As a reminder, the public webinar series is designed to educate attendees on the scientific ecosystem, and each month we'll have representation from a data coordinating center and new developments from the centers that support them. Today, I'll start off by sharing some KC scientific updates, upcoming events within the CFDE, and we'll hear from our featured speakers. If you were unable to attend last month's webinar, all of our webinar recordings are available on our YouTube page and available at this link here. We are currently actively working on site updates to the KC, which will include a new front page design as seen here. And we're also going to be updating the foundational knowledge pages. And we'll have two brand new analyses, the gene set analyses and differential expression analyses. And we're going to unveil these new updates soon. So if you're going to be attending the ASHG conference, we'll be able to show this off for the first time in a full setting there. But keep an eye out for that. If you have any feedback, questions, or would like to be added to our listserv, please email us at help@cfdeknowledge.org. So this week we attended the October 2024 CFDE face-to-face meeting in Bethesda. We were able to hear some updates from the various programs and centers, and we were introduced to the new Cloud Implementation Center and the Training Center, which are the final two centers of the CFDE. So we'd like to continue extending the warm welcome to these centers here at the new webinar series. As I mentioned, I was going to talk about some of the upcoming CFDE events that we have, and in just 2 weeks, maybe a little bit less than that now, we'll be attending a large conference in genetics and genomics, which is the American Society of Human Genetics. So we'll have two different events that will be going on during the ASHG sessions this year. First, we'll have our exhibition booth, which is November 6th through the 8th, and our booth number is 571. These are the different DCCs and centers that will be with us at the booth this year. We'll also have NIH there to give a perspective from the CFDE as a whole. Throughout the 3 days of events, we'll have different presentations and demos. So come back each day and there might be a new demonstration or event for you to attend. We'll be publicizing these as we get further to the event itself. And we'll also be hosting our ancillary session, A Human Geneticist's Guide to the Common Fund Data Ecosystem, which will be on Wednesday, November 6th from 11:45 to 1:15 p.m. in the Convention Center. We'll be going through the CFDE as a whole from the perspective of the NIH. We'll have the DRC present, and we'll share a scientific story from MoTrPAC and have some updates from the KC itself, and share some common fund opportunities from the NIH as well. So with that being said, today I would like to welcome the Molecular Transducers of Physical Activity Consortium, or MoTrPAC. Our speakers today will touch upon the MoTrPAC Data Hub, general MoTrPAC updates, upcoming data releases, and pipelines and database structure. So in addition to their consortium, MoTrPAC is one of the data coordinating centers here in the CFDE. And I know the team will touch upon the Data Hub, but I just wanted to share a screenshot of their homepage with their link, which is up above, if you want to follow along at all during the webinar. So with that being said, I'll turn it over to our featured speakers, and just wanted to give a reminder to everyone, please feel free to utilize the chat and question and answer to ask questions. We're going to hold off answering the questions until after the presentation, but we'll be keeping track of all of them as we go along. All right. So now I'll hand it over to Jimmy and David.
Hello, can you hear us? I hope so.
Yes, thank you.
Let me share my slides first before we start. Slideshow. Hopefully you can see the full screen.
Yes, that looks great.
All right. Let me minimize this part here. All right, so thank you very much for the kind invitation and introduction. It's our pleasure to be here. I'm David Jimenez-Morales. I'm bioinformatics lead here at the MoTrPAC Bioinformatics Center. With me is Jimmy Zhen, which also will talk. Is the infrastructure lead at the Bioinformatics Center as well. We are going to talk today about MoTrPAC. We are going to talk about this comprehensive, multi-studies, multi-omic, multi-tissue insights into exercise molecular responses. You are going to hear the word multi many times. So, but the first take-home message that we want you to take is that we are the consortium that studies exercise. Right? MoTrPAC, I want first to formally tell you that MoTrPAC stands for Molecular Transducer of Physical Activity Consortium, that our goal is to study the molecular changes that occur during and after exercise. And we aim to generate this molecular map of the effects of exercise and training. I guess that the question, you might be wondering, why are we studying exercise? And here you can see a list of all the benefits that comes with exercise. It's the possibly one of the best medicines that you can take. We would like to understand how physical activity protects against some of these chronic diseases that are listed on the screen, but also how it promotes overall health. So this is a multi-center study, many institutions all over the country that are part of this consortium. And that's why first we need to start by acknowledging all these people, and thanks to all the members of MoTrPAC, we are able to generate what we are going to show today. This is also a multi-omics study, and we are covering, I think, probably all the omics that are available. And this is also a multi-species study, yes, where we have humans and our neighbors from the last 10,000 years, right? So that they are also going to help with this.
Best friends.
Yeah, we're best friends. So we are also a multi-study consortium, everything about exercise. And with respect to, let me give you an overview. With respect to humans, we have multiple studies as well. The first one that I'm going to mention is the acute exercise study, which is done on a relatively healthy cohort of human individuals, and which are randomized to either be part of the endurance or the strength study or the control, and they are doing a single bout of exercise, and we do a number of biospecimen collection. Then we have another cohort of highly active individuals as well that are randomized to either do an endurance or a strength exercise. We will hear more about these details later. Another study is on these sedentary adults, which in addition to this bout of exercise, acute exercise, there are also a training component for 12 weeks. And also in endurance and training, right? So, endurance and strength training. So the biospecimens that we are collecting are blood, muscle, and adipose tissue for the adults. And we also have a study on pediatric, but this is only focused on endurance, both an acute study, but also a training component. And the biospecimen that we are collecting for kids is only blood. And then we have the rat studies, and here's a summary. Later, we will talk more about these studies on rats. Basically, we are going to study two groups to different ages. One are the 6 months, which are the equivalent to young adults, and then we have the middle-aged adults. And we are studying like acute exercise as well, like in humans, and also training and endurance training. And one of the key elements of studying rats is the biospecimen collection. We perform a number of analyses and trying to characterize the molecular profiles in many of the tissues, so we can study this response to exercise across all these tissues. That's the advantage of working with this animal model. And again, this is the overview. I encourage you to read this paper that we reference here, the Cell paper, where is the markup paper where we explain all the details about this project. So I encourage you to read that paper. So MoTrPAC is aiming to address key gaps in the field. Here's a summary of the main studies conducted, the multi-omics study conducted on exercise. And MoTrPAC is aiming to bring a lot in order to advance our knowledge on exercise. But let's begin with the first study that we have released, that is on endurance training. And there is nothing better than showing you the timeline and the study design, so you get an idea of what we did here. So we have obviously rats that were that exercise, and we are talking about 48 rats that exercise for well, not for 8 weeks. I will explain you how this works. We have the controls, and as you notice here, the timeline is in weeks, and each of these green lines represent the bouts of exercise that they did every day. After 1 week of training, 12 of these rats were sacrificed and all the biospecimens collected. And the rest of the rats remain running and exercising. After 2 weeks, we have another sacrifice point, and so far and so on. So as you might notice, the length of these bars are increasing, and this is because this is a chronic progressive training where we increased the inclination and duration and the speed of the treadmill. And also important to notice here is that there is a 48-hour rest or washout period. So that's why we have these gaps between the exercise bouts and the collection, the sacrifice time point. So because we want to make sure that we are only studying the effects of training, right? Something important to point out here, another detail that I want to provide is that this is how the data is collected. So, especially, I mean, the goal here is that you to understand the amount of work that is behind this study. After 1 week of training, these are the number of rats that are sacrificed at each time point. And for each rat, we are collecting all these different tissues. And for each of these tissues, we are doing all these sums. We are covering like transcriptomics, epigenomics, by 2 different chemical analysis sites, 2 different labs, one in Stanford, another one in Mount Sinai. We have proteomics doing, protein abundance, phosphorylation, acetylation done at the PNNL and the Broad Institute. And then we have a large panel of metabolomics assays. We have untargeted metabolomics done at the Broad Institute, Michigan, or Georgia Tech, and then we have the targeted assays done at Duke, Mayo Clinic, and Emory. So, but we didn't do all the assays to all the tissues, but here we are summarizing what it was done in each of the tissues for this particular study, and we, as you can see, heart and liver got all of them. And then we have other tissues that got, a lot of other different assays. We're talking about white adipose or skeletal muscle, kidney, lung. All that got a, important amount of assays where we were trying to characterize the molecular changes. So, and now I'm showing here what you can see on the screen is a summary of the number of training regulated features. Let me explain this. On the left, you see each of the rows represent assay from each of the OMs. We have the RBS for the methylation, ATAC-seq to characterize the chromatin accessibility. We have transcriptomics, we have proteomics, phosphorylation, so far and so on, right? And importantly, the background color of the numbers is representing the differential proportion of analytes. So as you can see, for example, if we take a look at the epigenomics part, we didn't detect that many changes, relative to the number of peaks that we detected. But when you look at the transcriptomics component, you see that there is a huge variety of responses depending on the tissue. For example, look at the adrenal gland. We have it was the largest number of differential transcripts that we were able to detect, and then followed by colon. And then you see the adipose tissues, white adipose, brown adipose. You see large number of differential transcripts, and then blood. And then we have other tissues that we barely detect any change, like for example, in some of the brain tissues, like in cortex or hippocampus. And this variety of responses is also captured in the protein abundance assay. But some of these results, I mean, everything was, this study has been published. It was published like a few months ago, as you can see in the reference in Nature. It was featured in the cover. It's something that we are obviously very proud of. What we can do is to give you, it's a huge paper, but we can give you a few of the biological findings and a few of the highlights. For example, with respect to the phenotypic changes after 8 weeks, of course, we detected an increased aerobic capacity, the VO2 max, approximately almost at 20% in both males and females. Interestingly, the body fat decreased only in males, while increasing in the controls. And the body weight increased in females in all time points, and no changes, no significant changes were detected in males. Then we had like thousands of shared and tissue and sex-specific molecular changes. And some tissue remodeling patterns were observed, especially in the immune system, in metabolism, and in heat shock stress response, and also in the mitochondria or mitochondrial pathways. And these changes that we observed were consistent with some of the changes that has been characterized in humans. Something very interesting were the sex differences of the effects of training, especially in adrenal gland, in lung, or in adipose tissue. Very interestingly as well, we found potential links between some of these training adaptations and some other diseases, like for example, the non-alcoholic fatty liver disease, or inflammatory bowel disease, or cardiovascular health, or tissue injury recovery related pathways. So the Nature paper was like the landscape paper, but there are a number of companions, and we want to give you a few also biological highlights about some other papers. For example, this paper that was published in Cell Metabolism. And a couple of things to comment about this paper. One is tissue-specific mitochondrial adaptations that we were able to characterize. Endurance training induced substantial changes in mitochondria analyzed across multiple tissues in rats. The response to training was, it varied depending on the tissue, highlighting the tissue-specific nature of the mitochondria adaptation. In muscle, we were able to characterize an increase in oxidative proteins, which are important for preventing age-related decline. The level, and in liver, we were able to characterize a shift in lipid metabolism, which is potentially driven by the increase in acetylation of metabolomic proteins. With respect to the disease relevance, it was very interesting to notice that to find that some protein networks that were, for example, upregulated in exercise was downregulated in some other diseases, like for example in diabetes or cirrhosis. For example, it was very interesting in the case of this particular protein, HSD17B10.
I remember that.
Yeah, which was identified as a central regulator in both diabetes and cirrhosis. For example, it was upregulated with exercise and downregulated in disease. Very interestingly, the knockdown of this protein elevated the oxygen consumption in a human liver cell line. And as you might imagine, this has implications for mitochondrial dysfunction and metabolomic stress.
David,
But these are not.
Can I pause you for 1 second?
Yeah, sure.
There's a question in the chat from a couple slides ago, I think. It's from Doug Keel. It seems peculiar that the skeleton is excluded from all the molecular assays. Is there some rationale for this, given bone's exquisite sensitivity to exercise? That's a good question. What are your thoughts?
The skeletal muscle, we are including skeletal muscle. Which one? Vastus lateralis is the one that we decided.
Skeleton bone. Yeah. So there's concerns about being able to process the bone samples at the throughput necessary for the study. But there are bone samples stored in a biorepository for future analysis.
Yeah.
Oh, very good.
Yeah. That was Matt Wheeler, just in case that you guys couldn't know.
Yeah, Matt moving, still answering questions. I love it. A question from Henning. Were the rats free to feed as much as they wanted, or were they restricted to calories?
I think that they were not restricted as far as I remember. I mean, they were controlling the amount of food that was provided, but I don't remember from the top of my head if what was.
Yeah, there was no calorie restriction. There was a period of removing the food before sacrifice that was standardized, but there's no calorie restriction during this training.
Okay, we can keep going. Let's go back to where we were. Yes, we were mentioning that you have all the publications available. The ones, all of them are open access. So please go to this page and download all the publications. And thanks to this study, we are building a number of resources which are available to the scientific community. Obviously, an important one is the data. You can download all the data through the MoTrPAC Data Hub. We will talk more about how to get access to the data in a moment. Jimmy will walk us through the MoTrPAC Data Hub, which are mostly making available the results, different type of results. But then we have all the data also available in public repositories. If you are interested in analyzing the raw data, you can go to these public repositories. We are also making available all the source code and software that we have developed. For example, so we are trying to make everything transparent, from data injection, all the code to for the pipelines that we develop for the identification and quantification, or everything was developed in close collaboration to each of the labs that are part of the consortium. We have source code for quality control, extensive data analysis, and also code available. Very importantly, we made available some R packages, and this is a very nice feature, and also something that should be pretty useful. So in most of our users in the consortium use R, so that's why we decided to put everything in R packages. And it's as easy as install that data library that we are listing here, and you will have all the differential abundance results and all the normalized tables for you to do your own analysis. But if you need some extra functions, we also built another library that helps you do some analysis on our data sets. You will be able to reproduce the results that we have in the papers, right? For example, I don't know, if you want to do a PCA, we'll help you with that in that library. They are nicely documented, so we encourage everybody to check it out. So there are more. So for each of the publications, every time. So we are trying to make our packages to facilitate the exploration of the data. Again, we want the world to use our data and trying to help him unveil and anything that could help us understand how exercise benefits us, right? And now I'm gonna Jimmy is gonna walk us through the MoTrPAC Data Hub.
Hello, everyone. Yeah, so my name is Jimmy, and I am the software engineer at the Bioinformatics Center for MoTrPAC. So what I'm going to do is to really talk about the MoTrPAC Data Hub, which is the primary resource that we have built for our users to access the data, to explore the data. So having that we already discussed or share with you, like what the data sets that we have available. So today, we're really going to just focus on the endurance train young adult rats study data that we have publicly accessible on the Data Hub portal. So the first thing I want to talk about is to really just dive right into like how our users can just access the data right out of the gate, right? So the first thing you want to do is to, like, when you come to the Data Hub homepage, or let's say just come to the Data Hub portal. So the first thing you want to do is to go up to the top navigation and click on the download button. And once you do that, you will be taken to the data download page. Right. So on the download, the way the data download page is organized is to give users access to a couple of different ways to access the data. Right. But before before we go any further, I really want to make sure that our users have a good idea what types of data or what type of information that we have included in the publicly accessible data sets, such as, obviously we include quantification results and sample metadata, QC reports, and differential expression analysis results as well. So there's quite a few things, and also want to point out that currently we don't have all the raw files available for download through the data portal due to their file sizes, but please feel free to reach out to us if you do want to have access to the raw files, and we can facilitate that and help you to get what you want. So feel free to let us know. Now, so in terms of downloading data, if you scroll up the page a little bit, you can see that we have assembled a number of pre-bundled data sets that we have designed to make it a little easier for our users to get the data that they are particularly interested in. So they are kind of organized the bundles are organized in ways that they are grouped by ohms or they group by tissues. So for the ohm bundle data sets, each one of them includes all 19 tissues from the endurance trained young adults. For the tissue bundles, they include all 5 ohms as well, and commonly they all include all the quantification results, the phenotypic data, as well as the differential expression analysis data, right? So, and now if you scroll back up, and let's say if there are if users are not interested in downloading a large bundle data set, but they really want to customize the data set that they're interested in, they can scroll back up and click on the Get Started button, and this will take you to the next page, which allows our users to really customize the kind of data that they're interested in downloading. For example, when you come to this page, you can use a number of different filters on the left. So for example, in this case, we have selected skeletal muscle, heart, kidney, and then we select the transcriptomics as our primary ohm. So once you have selected the filters that you are interested in, and you will see the table or the files on the right side will be automatically changed to match what your criteria are, right? And then once you're happy with some of the choices that you have made, you can select the number of files that you're interested in. Sorry, you can select the number of files that you're interested in downloading, right? And then you can go ahead and click on the download selected file button on the right. And once you do that, you will be prompted to enter your email address and name. So that the reason that we're doing that because that we are zipping up all the files that you have requested for download in real time, and then we will notify you when the zip file is ready for you to download. So that's why we're doing this. So yeah, so the download link. So when the zip file is ready for you to download, download link will be sent to you within 24 hours, and the download link will be valid for 7 days. So yeah, so that's how our users can get access to the data either through a customized a fashion or just download the bundles that we have pre-assembled. Right. So now the next thing I really want to talk about is to show our users how to explore the data in different ways on the data portal. The first thing that I would like to highlight would be the differential abundance data. So the way that you can access that is to go up to the to click on the differential abundance link in the Explore drop-down menu at the top. And once you do that, and you will be presented with a page that that looks just like that, except that in the beginning, you probably won't be seeing some of the search results, because that what our users would normally do is to first of all, they start with either a gene or protein ID or metabolite, right? And then they can enter either multiple genes in the search field, or as also at the same time, they can use auto suggestion support that we have designed so that our users don't have to remember or memorize the gene symbols or metabolite names, right? So, and then once you have the number of genes that you're happy with, and you do a search, and then you will be presented with search results, as you can see on this page. And then once you once you see the results, you can also utilize the filters on the left to sort of narrow down the search results into a smaller subset. Also please know that the search results or the analytes are grouped in two different tabs, either by the time point specific tab or the overall training tabs. So just want to make sure that our users are clear about about that. Okay, and then so let's say if you have also selected a number of different tissues or certain assays, like we previously talked about in the data download section, right? Once you have selected your filters, you can click on the update results, so you can get a new subset of search results. Once you do that, okay, and then you can also click on the download results button on the right, so that you can download all the search results that you're seeing on this page. So, so the next thing we also want to highlight would be the gene-centric view, and you can access that in the same Explore drop-down menu as well by clicking on the gene-centric view. And once you do that, you would be presented with a page that looks similar to the differential abundance page, except that we have a smaller number of filters for users to choose from, because this is primarily focusing on the RNA-seq assays as well as the proteomics assays, right? So similarly, in here you can search, do your search against multiple genes, and we also have auto suggestion support for the search as well. Similarly, you can use the filters to sort of narrow down the search results in the smaller subset by updating the search results, right? And then after that, you can what you would want to do is to select a number of features that you're interested in, right? And then once you do that, you would click on the view time series plots button on the right, and when you do that, you will get to see the time series plot for all the features that you have selected. And you can obviously in this screen you're only seeing the three of them, but if you scroll up and down, you will be able to see the rest of the time series plots. So moving along, so another thing that it's quite essential to the endurance trained young adult rat study data would be the graphical clustering results. So you can access it again in the same menu by clicking on the graphical clustering link. And once you do that, you will be presented with a page that that we have all the graphical clustering results organized by the published MoTrPAC landscape paper as well as the mitochondria paper. So there are two groups of these reports, right? So if you scroll further down, and you will be presented, you will be able to find all the graphical clustering graphs that we have organized by tissue, and you can also you can use the jump menu to jump to a specific cluster or the graph that you're interested in, right? So once you and then you can scroll up and down and see the a detail of all the different graphs for the different clusters. Now, we also have built an interactive data visualization tool to supplement or to complement this graphical clustering results, and you can access it in different ways. Obviously, you can also go up to the Explore drop-down menu to access that, accessing with the link. And also, you can simply just click on the Explore interactive trajectory visualization button, and this button is kind of available throughout the entire page. So you can click on any one of them to access that. So if you click on that, and you will be directed to the interactive data visualization tool that we have designed to allow users to do deep dive into the endurance training data, and with that, so this is designed to let obviously right now this tool is very specific to the endurance training young adult study data set, right? But so the way to use this because this tool is pretty comprehensive, there are a lot of things that they are not immediately obvious to our users. So I would like to take just a few minutes to sort of go through this in details. First of all, our users would be able to customize the graphs by selecting tissues that are of their interest, the ohms of their interest, and once they have selected that, they would want to go ahead and click on the update button to update the graph, and then the users would be able to also sort of like do a further customization of the graph by selecting the clusters as well as the time points, right? And then and with the additional parameters that you can access on the right, you can really just drill down to the data set, to drill down to a graph that you are particularly interested in. And then and furthermore, our users will be able to click on this button to download the graph as an image if you're interested in having that graph in either your presentations or in some of your research writings. So so moving on. So one thing that I also want to as I previously mentioned, that some of this the way that to use this graph may not always be obvious to our users. One thing that I do want to highlight is that in the parameters drop-down menu on the right, if you select show features, right, and then you click on any one of the paths, you will be able to see a highlighted path and see additional metadata about that path, which is something that's not always obvious to our users in the beginning, right? So, and then the next graph that I also want to highlight would be the cluster composition. Again, some of these features may not always be obvious to our users right off the bat. But if the users sort of mouse over the different bars on the graph, you will be able to see some of the tooltips showing you additional information. But if you click on any one of the bars, right, you'll be able to bring up additional mode or to that displays all the data relevant to that particular graph that you're looking at, and you can download that data as well. So these are the things that may not always be obvious to our users right at the beginning, right? So I'm going to go ahead and jump to the next one, would be the feature trajectory graph, which is also highly customizable by using the parameters on the right. And lastly, which is the pathway enrichment graph, which is also highly interactive because our users can pan around, pan a graph, from left to right, up and down by just dragging it, or by using all these control keys at the lower left-hand corner, or they can also zoom in, zoom out, and reset the graph, and also by dragging different nodes, and also by clicking on highlighting or mouse overing any of these edges or nodes or this callouts, then you will be able to see additional metadata about the graph. So I think that's pretty much a lot of things that I really want to cover in terms of the MoTrPAC Data Hub portal, as well as the data visualization tool, because this is kind of important to our users when they want to come to the Data Hub portal to access and find the data that they need, and to explore the data that they're interested in looking into. Now, and briefly, we really wanted to talk about is that without, although we have designed and built a lot of backend services to support all the things that we can do on a data portal. These are things that our users don't normally see because they're all kind of happening behind the scenes, but they are very important to to the daily operation of the data portal. They provide, they provide the need for us to scale this data portal, for us to make sure that everything that we do is maintainable, everything is performant, particularly the file download service, which is a very essential component for users to download the data, right? Particularly when they do the customized data set download. We have put a lot of thoughts into this service, and as you can see in the diagram that we have designed for the file download service architecture, right? And we also have put a lot of thoughts into architecting the search service as well, which is also an important piece, an important component for us to support the differential abundance search, as well as the gene-centric view feature on the data portal. So with that, I'm going to turn this back to Zhen. I think I might have to.
Before we turn it over, there is one question though. So there's actually two. So what is the license for the data? And then what's the update frequency? And do you provide a versioning?
So we don't really have licensing or restrictions on the use of our endurance train young adult rats study dataset, but we do plan to have a simple license. I forget, it's a CC4, which is a license that we encourage our users to acknowledge a consortium when they use the data set in their research publications. So that's I would say that's a pretty minimum.
Thank you.
All right. So with respect to the frequency, we are gonna finish with that to address the second question, is that about the upcoming data sets. And for that, let's bring back the timeline of the endurance training. As I guess that you all have in mind the complexity and the dimension of this data set. Now we are also studying the acute exercise, where instead of having these 4 time points for the endurance training, now we have all these 7 time points, immediately post-exercise, half an hour later, 1 hour, with all the controls, all the way to 48 hours. 48 hours will be our connection to the endurance training. This is a data set that is currently being analyzed. It should be released next year. And if this is, as we were mentioning at the beginning, this study is in young adults, but we are also doing the same studies, both the acute and endurance training in the 18 months old, which would bring this component of aging that is going to be also pretty interesting. And with respect to the human data, because I'm sure that many of the attendees might be wondering what is going on with the human data. So what we didn't mention is that MoTrPAC started recruiting participants in 2019. In 2020, something happened that made us stop the study. Is what we call the pre-COVID data set. So obviously it was interrupted because of COVID, and then 2 years later we were able to start all over again. So, but the consortium is currently working really hard in analyzing this data. I'm going to show you the same thing that we did with rats, is to show you the timeline of this acute exercise study in, again, these human sedentary adults. Which are, as I was mentioning earlier, a relatively healthy cohort of individuals that were randomized to either resistance, endurance, or the control group. Just for you to get an idea, and after 30 minutes in resting in our facilities all over the country, so we did the biospecimen collection of blood, adipose, and muscle. And then when they started the acute bout of exercise, we collected blood for the endurance group and the control group. Then right before finishing, we also took a blood sample for the endurance and the control. And then in post-exercise, 10 minutes post-exercise, we also collected another sample of blood from the participants. Then in the between 45 and 45 minutes of post-exercise, we collected again the whole pack of biospecimens. And then after 4 hours, another biospecimen collection of blood, adipose, and muscle. And then the next day, they came back at the same time, they rested, and we took the final biospecimen collection. All these data sets currently being analyzed, and it should be released next year, or better say, is going to be released next year, hopefully early 2025. I'm pretty sure that, so by the way, there is a summary of all the projects in the portal that you can visit. And as you were, our attendee was asking, and of course, if you want to be notified about the upcoming data sets, you can do that. You can go to the portal as well, to data access, and go to the data update signups. Click there and you just fill up the form, and we will inform you as soon as the data sets are available. And there, more about MoTrPAC, you can learn more about the consortium by visiting the consortium website. What we have talked today was about the data portal, but there is a consortium website with lots of information. You can also visit our YouTube channel, where we are starting to put together videos where the tutorials, etc. For example, right now there are a tutorial for the data hub in 2 languages. We are covering Spanish.
Yeah, we did
English.
the whole Spanish.
Yeah. And then we have also open office hours that you can sign up. They are bi-weekly. You can come with your questions. We are building a community with Discord. And by the way, if you can also join us. We are at the American Society of Human Genetics. We are going to have an ancillary session where we are going to go through the portal, but also through the R packages. And we are also going to be part of the Knowledge Center workshop the same day. Then we also have, we are going to be present in the Integrative Physiology of Exercise conference in Penn State. So we will be there. And finally, we would like to mention our collaborations with other Common Fund members. This is an exciting opportunity. We have several collaborations right now. For example, the Transcriptome Variability Partnership is how you can leverage data sets from multiple consortium. We also we are starting to work with Metabolomics Workbench and other consortia to do the metabotype. And yeah, you can find more about us in the Common Fund resources. And yeah, of course, again, we need to acknowledge again everybody from the consortium. Without all these brilliant people who have all these resources that we are building. Of course, we would like to thank you all. Matt can say a final words here.
Yeah, so I want to especially thank the team here at Stanford to help manage our Bioinformatics Center, our back-end development, and our front-end that you've seen today. And certainly both our current members and our alumni from the MoTrPAC team are integral to the work of MoTrPAC, and we're excited to be continuing to be a part of the Common Fund Data Ecosystem team as well. So I think with that, we'll take your questions, and again, appreciate David and Jimmy doing the hard work today.
Great. Thank you. And so as Matt was saying, feel free to ask any questions, and we can also allow you to speak if you raise your hand if you want to ask a question over audio instead.
Yeah, I think there was one of the prior questions around versioning was asked, I think. So in terms of the publicly released data sets, we do have an anticipated version release will be coming probably early 2025 that would include remapping of many of the analytes to newer versions of the rat genome. And then with the human data coming in, there will be enhancements to some of the visualizations and search as well. So we are typically doing portal updates on a quarterly basis, typically near the end of the quarter.
Any other questions?
All right, well, if anything is not crystal clear, feel free to reach out to us at the MoTrPAC data.org help desk, and we can address those issues as they come in.
Awesome.
Thank you to the Knowledge Center team for hosting this.
Great. Well, thank you so much for participating in this event. And again, reminder that we'll both be at ASHG. So if you have any other questions and you want to talk to us in person, please feel free to come visit us both at the ASHG booth and at the 2 ancillary sessions that we'll be having on the 6th. And just before we head out, our next webinar will be on Friday, November 22nd. And I'm just highlighting this specifically because this is not the last Friday of the month this time, because we want to accommodate for Thanksgiving. So please keep an eye out for that schedule change. And thank you so much again to the MoTrPAC team. We really appreciate having you here.
Thank you.
Thanks, everybody.
Thank you.
Enjoy your weekend.
Thank you.
Bye-bye.
Bye.